Here we're going to discuss DNA sequencing. But in order to get started on this topic, I need to talk a little bit about a process known as DNA annealing and DNA denaturing. If you had two samples of DNA, say sample 1 and sample 2, and you were to heat them up, the heating of the DNA causes it to denature. And when DNA denatures, basically it goes from double strand into And when DNA denatures, it goes from a double-stranded molecule into single strands. Now, if you have two samples of DNA which you have heated and they are now single strand, and you mix them together and you cool them, then if these DNA molecules have regions of similar sequence, then when you cool them, they will come back together, a process which we refer to as annealing, and some of the molecules will come back with their original partner, but some of them will anneal with the DNA strand of the other sample. And so you'll get duplexes of sample 1, duplexes of sample 2, but then you'll also get hybrid duplexes in this annealing process. And this is an important process in order, for us to understand in order to go forward in our discussion of DNA sequencing. In order to understand DNA sequencing, it's best to start with trying to understand DNA synthesis. So we'll discuss that real briefly here and then move on to showing how that applies to sequencing. So in the synthesis process, you basically have a template strand that guides the incoming nucleotides, or it determines the identity of the incoming nucleotides. You have a primer strand, and the key characteristic of the primer strand, other than it's complementary to the template, is the 3' hydroxyl. In DNA synthesis, and this is true of RNA synthesis as well, new nucleotides always get added to the 3' end because the 3' hydroxyl adds as a nucleophile that will attack the phosphate of the incoming nucleotide and form a phosphodiester linkage. The identity of the new nucleotide is governed by the template strand. Now, if you were to have what is referred to as a DD-NTP analog, and that is a dideoxy nucleotide triphosphate analog, the key identity here is that it's lacking a 3' hydroxyl. So if this were introduced into your synthesis mixture and you were to have this come in, then it would get added because if the previous nucleotide had a 3' hydroxyl, there would be a 3' hydroxyl to perform the reaction. However, this one would be absent, and so any incoming nucleotides would not be able to form phosphodiester linkages with this strand. It would effectively be truncated and would become a fragment. Okay, it would not be able to extend. It would be a truncated fragment. Now, let's take what we've just talked about and combine it together in a different sort of figure. Now, in order to carry out DNA sequencing, there actually is an older, more traditional method, which we're going to talk about here, and there's a newer method, which we'll talk about later. But I think the old method actually illustrates the process slightly different way, but it illustrates it a little bit better because it allows you to see the different bases and how you're generating fragments for the different base reactions. So let's go through this. Now, in order to carry out DNA sequencing, you need a template, you need a primer, and you also need a mixture of all the normal nucleotide triphosphates. And for DNA, that's going to be deoxy nucleotide triphosphates, A, G, and C. Okay, so you need a mixture. And then, of course, in addition to this, you would need the correct buffer in order for the enzyme to be active. So into this mixture would go enzyme and buffer. Now, once you have this mixture, you then separate it into 4 separate reactions. These 4 separate reactions you label A, C, G, and T. And into reaction A, you put dideoxy ATP, and then into C, you put the C version of that, and so forth. Now, let's just focus a little bit on one of these reactions and see what you get. Well, if you start off with a template and you have a mixture of the normal NTPs, well, you're going to be able to synthesize an entire complementary strand from the primer. However, once you add in a small amount of this, some of the time you're going to add in a dideoxy-A and you're going to get a truncated piece of DNA, or you're going to get a fragment. Okay. Now, let's suppose you put a radioactive label on the end down here on the primer somewhere, like say the 5' phosphate, for example, and that radioactive label will actually light up on a gel. It will, if you take and run these fragments on a gel and then expose the gel to um, X-ray film, then wherever there was radioactivity, you'll get a spot on the film. And so when you run out this mixture, this reaction mixture, you get 3 fragments. The 3 fragments correspond to a small fragment down here. That was the primer that got truncated here in the second, where the second nucleotide got added, and it was a dideoxy-A. And then you have a second fragment and then a third fragment. Now the third fragment corresponds to the primer coming all the way out here to the very end. So it's the longest fragment. It moves the shortest distance down the gel, starting from the top. It moves the shortest distance. It is the longest fragment, and it corresponds to the 3' end of this primer. Now, if you did the same thing for all of these other samples, all 4 samples, you do the same thing, and you run out each sample in a separate lane, you get an A lane, a C lane, a G lane, and a T lane. Now, in order to determine the sequence then, you have to combine all of this information. And the way that you would do this is you would just start at the top of the gel, and you would say, okay, my first base is, my longest fragment ends in A. So A is at the very end. My next longest fragment ends in G. So G is the next nucleotide working backwards toward the original primer. So I would go A, G, T, and I come back over here and I get a C, and then I get a G, A, G, and then I pick up a C. I've got 2 T's in a row, and then I've got an A, and then at the end a G. Okay, and that's how I would build the sequence, and I would just write it out here. Now, what is this sequence? Well, it's the sequence that corresponds to what gets added onto the primer, so it's the complementary sequence of the template. Not the exact sequence, but the complementary sequence. And at the very top of this is the 3' end. That corresponds to the 3' end of the fragment that ends up here. And then we're basically getting the sequence as we go back toward the primer and as we come down this way. Now, to get the original sequence, we would have to take the reverse complementary of this sequence, right? And how will we do that? Well, we would just write over here 5', and then under 5' we would go under 5' it would be T, because it's complementary to A, so it would be T, C, A, G, C, T, and so forth. But it would be 5' and then the sequence that is complementary to this. And that's how we would generate the reverse complement. And actually, that's what would be the sequence of the original template.